transformation of E. coli is such a ubiquitous experiment. It's done at the high school levels, it's done at the college levels, it's done all over America. It's done in several classes here at Sinclair, different classes. Microbiology does it, we do it. So here's the problem. So many students have written so many reports on this and turn it in as receive these, that there's the similarity reports are going to be a little higher sometimes in some cases. Now, that doesn't mean that students don't get low similarity reports, but some students will start to sneak up there a little bit. So I will expand it from having to be below 20% to having to be below 30%. And here's the issue. What will happen is it will start nailing certain combinations of words, because there's been so many of these reports submitted. It'll nail a sequence of words in a variety of different paragraphs, and that will add to your similarity report. If it's doing that, that's not a big issue. Plagiarism is when it nails whole sentences. Whole sentences are highlighted, or whole paragraphs are highlighted. When you guys look at your similarity reports, you should be able to see where it's highlighted things, correct? Has anybody gone in and checked when they saw the, well, maybe you saw a low score.
Yeah, like for the natural selection paper, the only thing that I got for the similarity score was the title page. Like when you have the title page, except my name, everything else was highlighted.
Yeah,
but that was the only thing that I had.
Okay, well, here's the point. When you guys submit your transformation reports, check to see what's highlighted. If it's just highlighting a couple words here in a sentence, a couple words there, you know, if it's just got spotty highlights, that's not an issue. But if it highlights an entire sentence or sentences or paragraphs, that's where you're going to have to rewrite. Does that make sense? So I will go up to below 30% on this one. The rest of them will stay at, the other papers will all be below 20%. And you guys will, so you probably want to submit it, you know, it's due September 11th, but you have until 11:59 that evening. So you have time to submit it and check your similarity reports. Questions about the other thing about the transformation report is I will promise I'll have it done by the lab exam. Usually I try to turn these around quickly. So for example, your natural selection papers, I knock those out pretty quickly. Those are a little easier to read. These will be harder, but here's my problem. I have 3 evolution classes and I'm getting papers in those classes. So I'm getting 70 evolution papers coming this Sunday. So I'm going to be swamped with papers. So my life is not going to be fun for the next couple of weeks because of the videos I got to produce, blah, 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 in these papers. So I apologize. You guys are not going to get your transformation reports back that rapidly. You may not get them back until right before the lab exam because of all the other work I have to do with other papers in my evolution class. So I apologize for that. But it's just kind of a combination of what's going on. I'll be busy doing videos and busy reading papers. Such is the life right now. Questions about the transformation lab report?
When you say
sorry, go ahead, please.
When you say you want the introduction to be 2 pages minimum, are you expecting us to go all the way to the bottom? Because I just finished my introduction and I'm like 1 paragraph onto the next page.
It has to be, when I say it has to be a minimum, who's asking this?
This is Drew, sorry.
When I say it has to be a minimum of 2 pages, you have to go, I say 1-inch margins. So when I say you got to go to the bottom, minimally, it's got to be 2 full pages of typed, double-spaced, 12-point font, Arial, 1-inch margins. And so you're allowed to go beyond that. You can make it 2.5, 3 pages. There's no maximum limit. Am I
Right.
explaining this right?
Yeah.
It's just if, let's say, and a lot of students, well, not a lot, but students do this. Let's say you turn in 1.5 pages. Well, you're going to lose 25% of whatever I determined the introduction to be worth.
Mm-hmm.
If the minimum requirement is 2 pages for the introduction and you're turning in 1.5 pages, that's only 75% of what you did. So you're losing 25% of the points that I've determined the introduction is worth.
Right.
And sometimes I may not just make it mathematically logical. I may just take more points off because I'm not happy. I've told you guys, if you don't meet the minimum, expect to lose a lot of points. I can take as much off as I want. Meet the minimum. We're trying to get you guys ready to be hired by people someday, and places that pay well with good insurance and good benefits. Don't expect, they're not going to accept less than the minimum. Sorry.
Thank you. And then also, I feel bad bringing this up again because we've talked about this so many times.
That's
but
okay.
I still cannot find my similarity report and my percentage. I went in again for the natural selection paper that we did.
Yeah.
Could you just show me again, and then hopefully maybe I can.
Right. Yeah. Can you see my arrow?
Yeah.
My black arrow.
Mhm.
Go to progress, and then click Dropbox. And then you should be able to, I don't know if I want to click on these because I don't think.
Drew, when you do it, when you drop it into the natural selection or whatever it was, click on the natural selection, and then at the bottom it'll have
You want me to click on
turn it
natural
in.
selection?
Yeah, that's what we have to do. We have to click
Yeah.
on it.
You have to click on the natural selection?
Yeah, and then we turn it in through there, and then on the bottom, it'll tell you your similarity score. And then from there, it can give you a link that you can go into the Turnitin website or whatever it is, and then it'll have it highlighted what you have that's similar and whatnot.
It's weird. So I clicked on the natural selection, and then it takes me to, because I did submit it. Like I submitted it both ways after we talked the last time.
Yeah.
And when I went in here, it's just giving me an option to submit another file, and it has a little thing.
You have to go to for it's different on the professor side. For the student side, you have to when you click on the Dropbox, you should see so there's the natural selection, and then to the right there's another column that said completion status, and then it should say, if you already submitted it, it should say 1 submission, 1 file, and it's highlighted. So and then to the next of that, there's a score. So you click on the 1 submission, 1 file. You get to the page where it tells you the report. There's like a column that report, and there's a percentage right there. That's the target percentage. And then
are you following this, Drew?
Yes, I see mine now.
Okay. You see how to get it?
Yep.
Okay. Anybody else have questions about the similarity report?
So I have a question about the lab. Are we still on?
We're still on the transformation.
Yeah. So the title page is APA format, correct?
Yeah. So, yeah, but use the format here. Just go there, click on that, and follow this. I thought that there was just a standard APA format, but apparently there's variations of this. Let's use this one. All I want is, to be honest with you, I don't care if it's done, but I've picked this one. So you guys have to use this. This is the standard we'll use in this class. Just follow this. And where it says affiliation, just put in there Sinclair College or Sinclair Community College. Okay? Just follow this. Yeah, follow this title page. All right. Other questions about transformation?
I'm sorry. I didn't catch what you said. You said that something about 2 pages. You said the introduction of this lab has to be 2 pages, or are you referring to the natural selection paper we did? That was 2
No, we're talking about the transformation report.
Okay.
The transformation, the introduction has to be a minimum of 2 pages, just the introduction alone.
Okay.
And then the other one was the discussion. The discussion has to be a minimum of 2 pages. And then I gave you guys flexibility on the other sections. I didn't put minimum requirements for the materials and methods and the results section. But you had a minimum 2 pages for the introduction and a minimum 2 pages for the discussion. And remember for the title page, you don't put the subtitle title page, but for the other portions of the lab report, you do put subtitles. You'll say introduction, you'll say materials and methods, you'll say results, there'll be these subtitles, discussion. Okay.
Some of us were just a little confused on where you want us to put where the pea glow plasmid and how it glows, the process of it. You wanted that in the discussion part, right? Not the
Absolutely.
introduction?
Yeah.
Okay.
Yes. The appropriate spot for explaining the regulation of the green fluorescent protein gene, which I'll take off a ton of points if I don't find it. And the location is in the discussion when you're explaining that plate, when you're explaining that the plate that's glowing, right?
Yeah.
It glows green. That's where it should be. Yep.
Okay. Thank you.
Yep. And then, as you guys know, we're going to start we got your first lecture exam coming up here. Not this Friday, but the following Friday. After that, you have your first laboratory exam. After that, you have a second lecture exam. Then you'll get a break. Third lecture exam. You'll see that we'll start testing here. We started off rather slowly, maybe, but we're going to start hitting the testing formats pretty often. All right. Any questions about that? Today's class is about this lab on population genetics. And what we'll do today will be tested on lab exam 1 and lecture exam 1. So there's a lot of overlap here between what we would have done in the lab for the population genetics experiments. So it would be beneficial for you guys as you obviously prepare for lecture exam 1. After lecture exam 1, you'll start preparing for lab exam 1. And you'll want to remind yourself, you want to go back to these lecture videos on the population genetics introduction, Hardy-Weinberg equilibrium, causes of microevolution, genetic drift, these here. You'll want to remind you. So the nice thing is, as you prepare for lecture exam 1, you'll also be preparing for lab exam 1. The two go hand in hand. So these videos through here, the lab exam isn't going to deal with the evolution of finches, the finch's beaks, or, sexual selection or things like that. But these first couple of videos here would be helpful to review before the lab exam 1. So let's get started. At this point, hopefully you guys are through chapter 22. You need to start working on chapter 23 now, if you haven't already done so, to keep up to get ready for lecture exam 1. Today's lab is about population genetics. And you can see in the name, population genetics, what we want you guys to understand is what is going on in a population of animals or plants. So let me review some of what's in the video that you guys have to do. What we mean by a population is a group of individual living things, a group of animals or a group of plants that are of the same species and they are living in the same geographical area, and they have the potential to mate. So a population is a group of individuals of the same species living in the same geographical region that have the potential to interbreed and produce fertile offspring. And what we want to do is we want you guys to understand some of the genetic basis of populations, because it is alterations in the genetics of populations that cause them to evolve. As you guys know, genes produce proteins. Genes are the genotype, proteins are the phenotype. If you're going to alter the bodies of animals or alter the bodies of plants, it's going to begin with the genes and then manifest itself in the proteins, which is the phenotype, right? So understanding what goes on with the genetics in populations helps us to explain why they evolve. To say that you're in the same species means that you are reproductively compatible. We have a whole chapter on speciation, which is chapter 24. But if we say that you're in the same species, this means that you're similar enough in how you look, in how you behave, in how you smell, and in your mating game. You're similar enough that you have the potential that you would have sex with each other. And if you had sex with each other and mated, you could produce fertile offspring. So if two organisms mate and they produce infertile offspring, they're not in the same species. The definition of species that we will use for our course primarily is one of reproductive capability. So if you're reproductively incompatible, if you can't produce fertile offspring, and that just means offspring that they themselves can reproduce themselves, right? They can go ahead and have sex and pass on their genes. If you're infertile, that means you can't reproduce. So that's what we mean by species. You're similar enough that you're capable of producing fertile offspring. So populations are groups of individuals of the same species living in the same geographical area. You can have various populations of the same species, right? Various populations distributed into different geographical areas. And species is the broadest term. Species is a group of populations, right? Of these, I should say species is a more broad term. Species refers to a group of populations living in different geographical regions that have the potential to interbreed and produce fertile offspring. Make sure you guys recognize that in the videos, I will mention many times that what's the smallest unit? Anybody know what's the smallest unit that can evolve?
Population.
A population, yeah. Individuals do not evolve. So the smallest unit that can evolve is a population. Species can evolve, but the smallest unit that we discuss here is that can evolve is a population. And often students will make mistakes on that. They will think that individuals can evolve. And that's just not what happens. You guys will want to, you can see here, you'll want to watch the videos on the Hardy-Weinberg equilibrium. Chase had a good question, which started the class off. In this lab report, I might change this first statement to from very large to extremely large population. I won't address the details of Hardy-Weinberg equilibrium here. I did so in the video, but here's the point. What they tried to do here, because some students are confused, why would you even have a concept of Hardy-Weinberg equilibrium if it never exists in nature? And the point is it's a theoretical concept to try to show you that evolution is constant. So what they've done here is they've tried to establish the conditions that would need to be met in nature for a non-evolving gene pool. And the only thing I'd change with statement 1 is from very large to maybe extremely large population. But if you see the wording of very large, don't let that bother you. Very large, extremely large, same thing for our purposes. But as you can see, all of these conditions have to be met in nature for you to have a gene pool that's not evolving. And these conditions can't be, in fact, it's not just one condition that has to be met. You have to meet all 5 of them. So look at condition 3, no mutations. Well, that's an impossibility. So right there, you can see this isn't happening. Now, as you'll see in the video, in some interesting cases, you can get isolated gene pools. So statement 2 here, an isolated population, that can occur. But in order for you to have a non-evolving gene pool, you have to have an isolated population that is with no mutations, with random mating and no natural selection, right? You have to have all these conditions met, and they just don't happen that way. It's an impossibility. But you might argue, well, why do you even go about doing this? Well, it establishes the fact that microevolution is a constant theme in nature, because in fact, the most fundamentally rapid way to get microevolution is to have an isolated population undergoing mutations, undergoing mate selection and natural selection, and that the population size is what? Large or small? What's going to cause a more rapid microevolution? Large population or small?
Small.
Small, right. So these are the conditions, and you guys, I'm going to have test questions on both lecture exam 1 and lab exam 1 about the conditions needed for Hardy-Weinberg equilibrium. And another way of saying Hardy-Weinberg equilibrium is genetic equilibrium. So sometimes in a question, I'll just say genetic equilibrium. And I also mean Hardy-Weinberg equilibrium. So it gives us a basis for comparison. The fact that you could argue maybe condition 2 here, isolated gene pool, that can happen potentially. I mean, it means that there's technically total isolation, and that might be difficult to get, but you can get situations where populations get isolated. But you can't get situations where mutations aren't occurring or where organisms are randomly mating. Mate selection happens in all living organisms, and you can't get situations where natural selection is not occurring. So because of the fact that you have to have all 5 of these conditions met for genetic equilibrium, and they can't be met, microevolution, and microevolution means genetic changes within the gene pools, is a constant fact of nature. Now, what's a gene pool? A gene pool is the totality of all the genes in a population. A gene pool represents the total of all the genes within that population, whatever population we're talking about, a squirrel population, plant population, whatever it might be. And it's not just the totality of the genes, it's all the allelic versions of all those genes. Remember what alleles are. Hopefully you did this in 1171. Alleles. Take the A in allele.
Yeah, the phenotypes.
What's that?
I said the alleles are the phenotypes, the phenotypic characters.
No, alleles are not phenotypes. The alleles, take the A in allele to mean alternate. Alleles are alternate versions of genes. So alleles are part of the genotype. Alleles make the proteins, which become a part of the phenotype, right? Does that make sense? So if we look at my skin color, I'm a little pale. That's because of the pigment that's being made due to the genetics that I have. So my phenotype of pale skin color is due to the genes that I inherited. That's the genotype, right? So alleles are alternate forms of genes, and they are part of the genotype, which will then determine the phenotype. Phenotype is the outward expression of the genes. We good on that? All right. Before I go further, are there questions about Hardy-Weinberg equilibrium?
Okay, so the Hardy-Weinberg equilibrium wants there to be a very large population for.
I don't know if we want to say it wants it to be, but what they're trying to say is if you're ever going to have a situation in which you get genetic equilibrium, you have to have a very large population.
Okay, but like microevolution occurs when the population is really small.
Well, microevolution occurs all the time in all population sizes, but it will occur the fastest in small populations.
Okay, I just wanted to clarify that.
Yeah, does that make sense?
Yeah.
Microevolution occurs all the time in all different population sizes, but it will occur more slowly in large populations and much more rapidly in small populations.
Okay.
Other questions? Now, guys, this isn't your last opportunity to ask. You know, next week as we'll get prepared for lecture exam 1, we don't have any lab activities next week. So we will finish population genetics lab today. We'll finish it, well, not today. We'll do it today. We'll finish it up on Friday. And then Monday and Wednesday, we won't have any lab that we'll be teaching. And you'll have plenty of opportunity to ask questions as you get prepared for the exam on Friday. Does that make sense? But once again, for any other questions about Hardy-Weinberg equilibrium, please don't misunderstand the point. Hardy-Weinberg equilibrium means genetic equilibrium, and it's something that never happens in nature under any circumstances.
I have a question.
Yep.
I was gonna wait until the end of class, but I can ask it now because it's kind of goes back to what you said about how you define alleles and genes.
Yep, alleles are alternate versions of a gene. So
Yeah.
that means that, let's take eye color, for example. There's eye color, the color of your eyes is actually controlled polygenically. So there's several genes that control eye color. But
Right.
for each one of those genes, there's several versions of the gene called alleles. So alleles are alternate versions of a specific gene at a specific locus. A locus meaning a spot on the chromosome. And you might say, well, why are there alternate versions of genes? Because mutations make them. Mutations alter nucleotide sequences, creating diversity of genes. And so controlling your eye color, let's say there's 3 genes controlling eye color. That would mean there's 3 locations on, let's say, separate chromosomes. So 3 different chromosomes have a gene that affects eye color. But each one of those genes is going to have alternate versions of itself called alleles. Now, for a particular gene, how many alleles could you have for one gene? What's the total number you could have?
I think it's 2 or more
You're
than 2.
You're diploid, right?
Yeah.
So here's the point. There might be, for a particular gene that affects eye color, there might be 6 different allelic versions in the population. But you, since you're diploid, could only have no more than 2 of the 6. If you have 2 different alleles, you're heterozygous. if you have two alleles that are the same, you're homozygous. This goes back to 1171 stuff. But in the gene pool, there might be six different versions of that gene circulating among individuals. But you, as one individual, could only have no more than two versions of it because we're diploid, we're not triploid. Does that make
So
sense?
every set of genes, since we're diploids, I think you talked about this in one of your videos, but we as mammals or animals, we have a complete set of genes, and we have two copies of the same gene.
We have two complete sets of genes. We got a set of genes from our mom and a set of genes from our dad, right? And the question then becomes, are the two sets of genes from your parents the same? In some cases, you compare the allele you got from your mom and the one from your dad, and it's homozygous, you're the same. In other cases, you compare the allele from mom and dad and they're different, then they're heterozygous. So yeah, for each gene, and what we mean by gene is we mean a specific location on a chromosome. And when we say locus, that means a spot on the chromosome.
Okay, so the gene refers to the specific location, whereas the locus refers to the spot on the chromosome. Is that correct?
Yeah. I mean, imagine a chromosome and genes are in specific areas along the surface of that chromosome. And when we use the term locus, L-O-C-U-S, that means the location on the chromosome of where that gene is. And you can have alternate versions of that gene. So if you look at the chromosome you got from your dad and compare it to the one from your mom, if you're looking at genetic material that's of the same gene, they'll be in the same spot on those two chromosomes, even though you got
in the same species.
Yeah.
Because it's different. The number of chromosomes is different from species to species, so you can't compare.
We're talking about the same species. Yeah, right. Yeah. But you'd be surprised if you compared our chromosomes to chimpanzee chromosomes. We're not in the same species, but we're so similar. You know, if you share 98.5% of your nucleotide sequences, you'd be surprised how many genes are in the same spots on the chromosomes. You'd be surprised at the similarity between chimp chromosomes and human chromosomes. But I get your point. There'd be some differences, but you might be really surprised at the similarities.
Oh,
right?
There's no doubt about the similarity. There's no doubt about that.
Right. And that's simply, all right, you follow?
Yeah, so a lot of times I hear DNA is described having each DNA has a location, a content. So the location is referring to the gene or the locus, and the contents referring to the alleles, I'm assuming here.
All right, how can I explain this?
Because the genes.
All right, I got the analogy. Listen.
Yeah.
You got a deck of cards. If on your mom's chromosome, if you got a king of spades, right? On your dad's chromosome, you got a king of hearts. Those are alleles to each other because they're different versions of the same thing. They're both kings. You'd be heterozygous. So we refer to them, alleles are just alternate forms of that particular gene. We would consider the king of clubs and the king of hearts to be alleles of the gene called a king. But you would not call the king of hearts an allele of the queen of hearts because the queen of hearts is a different gene. Does that make sense? Yeah. The queen of hearts would also be located on a different spot on the chromosome. So kings have four, right? You know, it's been a while since played cards, but there's four different versions of kings. King of spades, king of clubs, king of hearts, king of what? Diamonds? No. What's
I
the other
don't
one?
play cards. I don't know.
You don't play cards, but you understand you see my point here?
Yeah, I see the analogy, Professor. It's just a little confusing because molecular geneticists they oftentimes use the word alleles instead of genes. But
Well,
it
okay,
seems the way
okay.
you're describing to me that the
Yeah,
two
so-
are both
so here-
having locations.
So here's the thing. All alleles are genes. All allele is just an alternate form of a gene. So all alleles are genes. It's just that you can't say that all alleles are allelic to each other. Alleles, if we say we have a group of alleles, that's just the alternate forms of the same gene. So that's why I said if we're looking at alternate versions of kings, the gene is the king gene. And then the question is it the king of hearts or the king of diamonds? Those would be the alternate versions of the king gene. A king of diamonds is still a king. It's still a gene, right?
Right.
But if you compare the king of diamonds to the queen of diamonds, you cannot call those two alternate versions of each other. Those two cannot be called alleles because you're now dealing with entirely different genes. The queen of diamonds is a different gene. You can't say the king of diamonds and the queen of diamonds are the same alleles. They're not even alleles of each other because they're just different genes. And if your different genes are going to be located at different spots on the chromosome. There's a chapter in 1171 that deals with you guys might want to review, get back to like meiosis chapter. Yeah. Does anybody have anything to add on this? I think that analogy works. Well, here's another analogy. If you have socks that are identical socks, but they have different colors, the different colors of those socks would be alleles of each other. But let's say you have different types of socks. Well, those different types of socks would be the different genes. You would only say that you follow me here?
Yeah, Professor. It's a lot of times people use the word gene and alleles interchangeably. So I was trying to clarify that.
They do, and they do, and I do the same thing. And there's a reason for that, because anytime we use the term alleles, all alleles are genes. They're alternate forms of a gene. But we can't say that. We can't turn around and then just say, hey, anybody got anything to add on this? It's silence.
It's just simple. You have two, one from your mom, one from your dad, and all of it together is the gene pool. Because like, think about the human population. You have all our genes together as a gene pool, but if you take you individually, you have two genes for a specific trait. So like for hair color, you got one from your mom and then one from your dad. That's what he's trying to explain, if that makes sense.
Right. And what we're saying is that you probably got a different version of the gene from mom than from your dad. And so those would be alleles, right? Alleles are the alternate versions of the gene. And hair color is probably controlled by many genes, polygenic inheritance. And so again, let's say there's 3 different genes. Those genes would be very likely at different spots. Well, they would be at different spots and maybe even on different chromosomes. And the alleles are just referring to the different versions of those genes.
Yeah, I understand that. I was just trying to clarify with the usage of the word
Right.
gene
So
and allele.
you're right. We do interchange them, and that's fine, because it is true to say that all alleles are genes. They are simply the alternate versions of specific genes. It's just not true to say that all alleles are allelic to each other, meaning if we look at the king of diamonds and compare it to the queen of diamonds, those are two different genes. We don't say those are allelic to each other because the king of diamonds is not an alternate version of the queen of diamonds. They're separate genes. Does that make sense?
Yeah, that's really interesting. When you say that a gene has 2, 3, 4, 5 different alleles.
Yeah, in some cases,
so
10 different alleles.
That's interesting because those alleles are located on the same location as that gene. Is that what you're saying?
Yep.
Okay.
Yep. If we say that you're heterozygous for a gene from your parents, they would be in the exact same spot on the two homologous chromosomes, and there'd be just a subtle difference in the nucleotide sequence that's causing the alternation, the alternate form of it. Right.
Yeah.
All right.
That makes sense. Thank you.
Yep. Yeah, that's all right. That's all right. But you know, Chase, you like to really think this stuff through. I would go back, and I think it's just the beginning of the chapter on meiosis. There's a whole section on homologous chromosomes. Just review homologous chromosomes and homozygous versus heterozygous conditions, and hopefully that'll explain it. And maybe you have it now, but if you don't, we can deal with this again Friday and next week too.
Yeah, thank you.
Yep. All right. So, Hardy-Weinberg equilibrium never occurs in nature. And there's a reason for that. And that reason is things like simulation 1 here. If we were face to face, we would have worked with these beads and we would have done experimental simulations. You guys, you know, you guys can do this at home. I'm not going to force you to turn in the data to prove to me that you did at home, but I would highly recommend that you go ahead and do these simulations. More on that in a minute. But here's one of the reasons why microevolution occurs. This first simulation is called gene flow. And gene flow simply means a situation in which members from one population are migrating into another population and they're having sex and moving genes. Gene flow is between populations. So be careful, guys. If I've seen any problem students have is they will think gene flow means flow of genes in a population. No, look at the definition here. I don't know why that can't. Yeah, I think you see what I'm trying to do. Gene flow is between populations. And when members exit one population, right? Emigration, E-M-I, right? G-R-A-T-I-O-N, and then enter another population, immigration, you have members moving between the populations. And if members move between populations, they're of the same species, they may mate with each other. And if they mate with each other, they'll reproduce with each other and genes are being shared between the populations. That's what we call gene flow. Gene flow is a change in allele frequencies, or to Chase's point, we could say a change in gene frequencies, right? Because all alleles are genes. They're just the alternate versions of the genes. And gene flow is a situation in which the frequencies of genes get altered due to the movement of genes between populations. Well, if you say, well, how do you get genes moved between populations? Well, the things that carry these genes are the living organisms, right? And so you're getting individuals leaving a population, emigration, EMI, and individuals entering another population, immigration, IMMI, which is causing the gene flow. Now, here's my question. Do you think that gene flow between populations will cause them to become more similar in time or more different in time?
Depends on the amount of gene flows being shared between the two populations. The more gene flow there is, the more similar they become.
Correct.
They will be more different.
What conditions are going to cause them to be more different, Washburn? More gene flow or less gene flow?
More gene flow will make them to be different because they are getting different characteristics from the other, right? No?
I see your logic, but it's not correct.
Oh.
Yeah, what happens is if you get more gene flow between the populations, is the populations actually become more similar in time. Because let's say that you got population, well, and we can prove this. Washburn, I want you to do this simulation. You guys can go buy whatever you like to eat. You guys can go and buy some M&Ms and do this simulation. You could go buy trail mix. Might be a little healthier because it's only got, it's partially M&Ms, but there's all kinds of stuff in trail mix. I love trail mix, peanuts, right? Big raisins, M&Ms, all kinds of stuff in trail mix. You guys go buy some food and do the simulation. And Washburn, what you're going to see is over time in this simulation, these two populations are going to become more similar in their frequencies. So let's tell you what we're doing here. Population, so what they're talking about here is they have you're going to use red beads and yellow beads. These red and yellow beads are going to represent alternate forms of the bead gene. So we have a gene that makes beads, and it has two alternate forms. It has an allele that makes red beads, and it has an allele that makes yellow beads. The red and the yellow beads are alternate forms of this gene that makes beads. They would not be alternate forms of a gene that makes marbles. You guys with me? So in this simulation, what they did is we started off with a population A that had a red allele frequency of 50% and a yellow allele frequency of 50%. We have this population in a bag, and we've put the red and yellow alleles in there for this distribution of 50-50. We have another population, B, but that population has a different allele frequency for the bead gene. Notice its red allele frequency is 80. How do you determine the frequency of an allele? You take the total number of the beads you're looking at, right? So if you're trying to figure out how did we determine there were 80% red beads, look at the total of beads in this population. We had 50. And 40 of the 50 were red. So you take your red, the number of red beads divided by the total, and you'll get your red allele frequency. Down here, count the number of beads of a particular color, divide by the total number of all those beads in the population to determine that allele frequency in the gene pool. That statement applies to all of these simulations. So in this population, population B, we have 50 beads, 40 of which are red. So we say the red allele frequency is 80%. The yellow allele frequency is 20%. You say, why 20? Well, take 10 divided by 50, right? And you'll get your allele frequency for yellow. So in this simulation, what we would have you guys do is take, let's say, what do we say here? Close your eyes and select 6 beads from population A bag. Close your eyes and select 6 beads from population B container, and then open your eyes and trade the selected beads. So what we do is we pretend that members from population A were going into population B, and members from population B were going into population A. And we would, each generation, so you take 6 from population A, move them to population B, 6 from population B, and move them to population A. And you would, so that would be one gene flow event. You'll see that if you do this 6 times, you'll start to see these allele frequencies becoming what? More similar to each other or more different?
Or similar.
More similar to each other. In fact, you'll start to see this red allele frequency start to go up, and you'll start to see this red allele frequency start to go what? Down.
Down.
Very often in class, when I see this, after about 6 generations, usually the red allele frequency is about 60% in this population, and the red allele frequency is about 60% in this population, roughly. You know, it might be 64% here, 62% here. But the point is, because of the gene flow between the populations, they are becoming more similar in the distribution of the alleles, and the allele frequency is becoming more similar. Now you say, well, so what? What's this matter? We're trying to explain to you guys why populations evolve. And then we're going to try to explain to you how we get macroevolution. So if you have gene flow between populations, this is causing microevolutionary change. But it's causing microevolutionary change, which will decrease the probability that these 2 populations will diverge enough to become separate species someday. So gene flow between populations decreases the chances of macroevolution in the populations, while at the same time driving microevolution. As we'll see, macroevolution, the first form of macroevolution, will be speciation. More on that later. I'm going to stick with an older definition of macroevolution. For macroevolution for us will be at the speciation level. Your textbook has gone to a modified form where it talks about microevolution. Then it talks about speciation being the bridge between micro and macro. It almost gives you a misleading sense that speciation is not macroevolution. I'm not going to use speciation as a bridge. We're not going to go with the definition in your textbook. In fact, your textbook has changed that definition. We're going with the older definition from Campbell. And that is, we will consider speciation the beginning of macroevolutionary events. So the formation of new species is the starting point of macroevolution. Well, if two populations are getting lots of gene flow and they're trading genes between each other, they're going to have microevolutionary change. In this simulation, the red allele frequency in population A is going to move up, while the red allele frequency in population B is going to move down. Eventually, if you keep trading these, they'll start to get very, very similar. You're getting microevolution, but at the same time you're getting microevolution, this microevolution is causing these populations to become more similar. And it will decrease the chances of these populations becoming separate species. So why is this important? Because if you flip this and you say, well, what if two populations don't have gene flow? What if we reduce gene flow between populations? Then that's going to lead to genetic divergence in time. If something separates population A from population B, so in our analogy here, let's say that we're no longer going to share the alleles with population A and population B. What's going to happen over time is an increased chance for them to diverge from each other. And mutation is an important way for this to happen. Imagine in our simulation here, imagine we get a mutation in the bead gene to produce a purple bead. But the mutation only occurred in population A. And the purple bead, if there's no gene flow with population B, the purple bead will not appear in population B. You with me? So we'll start to get gene differences. Population A will start to have purple beads as the individuals reproduce with that purple bead. If there's no gene flow of population A, it's not going to get that bead. Population B might get a mutation that creates a green bead. The point is, if populations have reduced gene flow, they have an increased chance of genetic divergence, and in some cases, they might diverge enough to the point where they will speciate. We have a whole chapter on this. And when they speciate, they're no longer reproductively compatible. And you guys might say, well, that seems like it's impossible. How could you ever get the formation of new species? Well, it happens all the time. And here's one simple way. If you have a reduction in gene flow such that you start to get genetic divergence, all it takes is a few allelic variants to maybe cause, let's say, a mouse population to smell a little differently. So let's say that we're population A and B here, mice populations. They've had reduced gene flow. And mutations are affecting the two populations differently. And in the mouse population B, there's a mutation that's starting to spread that's causing the mice to smell a little bit different from population A. That's going to, if these two populations ever try to come back to mate, let's say that few members move into a population for mating purposes, because they smell differently, mating's not going to happen. Females on the planet are very selective when it comes to choices about who they're going to mate with for reproductive purposes. And let's say that a mouse from males from the population B sneak on in here in population A, and they want to mate. The females are not going to be interested in them if they smell differently. Now, there's odor variations in population A for individuals, but we're talking about a distinct difference in how they smell. That simple thing can alter the mating game. And if females aren't going to select for mating with you, then you're going to have stoppage of gene flow between the two populations. We have a whole chapter on this. I have lots of examples that I'll do eventually, probably in videos. But the point is, you guys need to understand that increased gene flow between, and gene flow is always between populations. If you increase gene flow between populations, it causes them to become more similar in time as far as their gene frequencies. If you decrease gene flow between populations, it causes you to become more different in time. And you guys can prove this to yourselves if you do simulation 1. Get some M&Ms or get a trail mix bag or go buy marbles, or just get some red paper and some yellow paper and cut it up into little pieces of paper and do the simulation. You'll find that the allele frequencies will start to become more similar in time if you have increased gene flow. Questions about simulation A? Let me stop. Questions about simulation A? So lots of gene flow, increased gene flow between populations, cause them to become more similar in time. Decreased gene flow between populations, cause them to become dissimilar in time. And so it's interesting to then say that microevolution doesn't always lead to macroevolution, because gene flow between populations will cause microevolution that decreases the chances of those populations speciating. While decreased gene flow between populations will lead to microevolution that does increase the chances of divergence enough for new species. And when we have new species, we have the first beginnings of macroevolutionary change. Questions about simulation 1, gene flow.
Are we going to just randomly just pick up, or we're gonna just
Right. Yes. Yes. Right. In this case, you are, with your eyes closed, we are assuming a random selection. Right. Yeah, you're randomly selecting. It's just like saying this, Washburn. A simple analogy. If I got a pile of peanuts over here, population A, and I got a pile of raisins over there, population B, and I start allowing the peanuts and the raisins to mix together over time through gene flow, I'm eventually going to have two piles of a mix of raisins and peanuts, right? That's the simplest analogy I can give. Now, here we're, raisins and peanuts are quite different. In reality, raisins and peanuts would be different species, right? But we're just pretending, right? We'll pretend that our raisins and our peanuts are alternate forms, right, of genes in two different populations. And right now we've got population A, which is mostly raisins. We've got population B, which is mostly peanuts. If we start allowing them to gene flow between them, they're going to become an evenly distributed populations of peanuts and raisins. That's about as simple as I can get it. Did that cover it? Or maybe I should use beer as an example, right? I got a population of Yuengling over here and a population of Bud Light over here, right? If we start to exchange the Bud Lights and the Yinglings, we'll start to mix them up. All right, questions about gene flow? All right, so gene flow is a cause of microevolution. And if you reduce gene flow between populations, it can lead to macroevolution. Here's another simulation that we would have done. We would have done the founder effect.
Wait, if we reduce gene flow, it leads to macroevolution?
If you reduce gene flow between populations,
Okay.
it will increase the chances of genetic divergence between them, which will increase the probability that they may potentially speciate, and that is macroevolution.
Oh, okay. Okay.
Yeah. So increased gene flow reduces the chances of macroevolution. Okay, let me say it this way. Increased gene flow will cause microevolution between populations that will cause them to become more similar, which will reduce the chances of macroevolution for those two populations. But reduced gene flow, did that make sense?
Yeah, I did.
Okay. Other questions?
I have a question about microevolution.
Yep.
So in this instance, when microevolution occurs, it leads to like it hinders macroevolution. But is there an instance where it can lead to macroevolution?
Absolutely. That's what happens when you get reduced gene flow, right? What we're saying is if you increase gene flow between populations, that leads to microevolution, but it's the microevolution of this type that will reduce the chances of macro. But if you reduce gene flow between the populations, that will lead to microevolutionary change, which will increase the chances of macroevolution.
Okay, that makes sense.
So,
So either way, there's going to be microevolution, but
yes, microevolution is always taking place.
Now I'm following you.
Yeah, because microevolution is just changes in the gene frequencies. And so if you increase gene flow, that alters frequencies. If you decrease gene flow, that alters frequencies.
Okay.
So microevolution is always occurring all the time. The question is whether or not macroevolution is occurring. And our simplest definition of macroevolution is the formation of new species.
Okay.
Macroevolution is the formation of new taxonomic groups. And I'll just
Thank you. That made more sense.
I'll just tell you, your textbook, we're not going to use your textbook's definition. Your textbook's definition of macroevolution is anything, the origin of a new taxonomic group above the species level. No, and they call speciation the bridge between micro and macro. I think that was totally unnecessary to make that change. Most textbooks aren't doing it. We're going to go with how they used to define it more recently, and that is macroevolution is the origin of any new taxonomic group, not above the species, but including the species level. So we're not going to say speciation is a bridge. It is, but speciation is the beginning of macroevolution, the formation of a new taxonomic group, a new species. Questions about that? We have a whole chapter on that.
Thank you for the explanation. That helped.
But
Okay,
if it's
good.
increasing gene flow, the population will be more similar then.
Gene flow, yeah. If we say, if we just say gene flow, that means you're getting the movements of genes between the populations. The question then becomes whether it's increased gene flow or decreased gene flow, right? Increased gene flow causes them to become more similar. decreased gene flow causes them to become more different. If you're going to become separate species at some point in time, you got to become different. And that was my point of talking about how animals smell. If you get mutations in a population that alter how some of the animals smell, but the same mutations are not being shared with the other population, then they're going to evolve divergence in how they smell. That's going to affect the mating game. And they could simply speciate because one population doesn't like the way the other one smells. They're just not having sex randomly with each other. All forms of life take sexual reproduction seriously, especially the females.
I think the word gene flow is confusing to some people because it implies that it's only going one way, whereas gene flow means the sharing of genes between two populations. So you think of it like this: you have two glass of water with different mixtures. You're pouring one into the other, and you pour the other into the other one. Eventually, the two cups is going to have very similar mixtures.
Right.
So that's a very good way to think about it.
Yeah, that's why I highlighted between populations, right? Yeah, you're right. Gene flow is it's movement of genes between the populations. That's why I used the term. Both populations are having some members leave and enter the other populations, right?
Maybe I'll wait until end of class for this.
Okay. All right, so gene flow causes a microevolution. Here's another factor. If we were together, we would do a simulation called the founder effect. It's a type of genetic drift. And genetic drift, as you guys are going to learn when you watch the videos, genetic drift is chance changes or chance fluctuations in gene frequencies in a gene pool. When you see the word drift, kind of think that you're on a raft in the lake, enjoying yourself, and you're just going to just kind of randomly drift around, right? Pretend you're on a river in a canoe maybe, and you're not using your oars, and you're just going to let the river take you where you go. Drift, think drift, you're drifting randomly. You're not trying to control where you're going. Genetic drift is not natural selection. Genetic drift does cause microevolution, but genetic drift is random changes in allele frequencies. So use the word, when you see genetic drift, take the word drift to remind you, oh, I'm just drifting randomly. And this means genetic changes in a gene pool that are unpredictable. Natural selection is not random. We'll get to that in a little bit. But genetic drift is random changes. And there's two forms of genetic drift that you guys have to learn about. You guys have to learn about the bottleneck effect, and you have to learn about the founder effect. The founder effect is the one addressed in the simulation here in lab. But let me briefly explain the bottleneck effect. A bottleneck effect is what happens when there are natural disasters in the world. So let's say, we are out in the West, and right now there's a bunch of fires going on, and you have this beautiful forest that's got a nice population of squirrels that are well adapted to that particular environment. And the fire comes through and burns everything. Burns the trees, burns the squirrels, kills a lot of things. That's not natural selection. If you have an environment that does not experience routine fires and the fire comes through and kills things, that's not natural selection. That is a random, the creatures that are dying are the ones that were in the wrong place at the wrong time. It's burning the trees and the squirrels that are in the, just happen to be in the path of the fire. And bottleneck effects are a form of genetic drift. And what happens is, let's say you have a squirrel, you have a species of squirrels, right? And there's many populations. And let's say we look at population A and it's distributed over this forest and the fire hits 20%, kills 20% of the squirrels. Well, that's going to have an effect on the gene pool of that population of squirrels. You just lost 20% of the genes from the gene pool. And you lost them at random. The fire, it was just, those squirrels were in the wrong place at the wrong time. That's not natural selection. That's a form of genetic drift. And this is why, one of the reasons why Hardy and Weinberg said you had to have a very large population, because they were trying to figure out conditions in which natural disasters, they're trying to say, well, in a way, Hardy-Weinberg equilibrium requires that there be no natural disasters. You guys with me? But they were trying to argue that if you had an extremely large population, that maybe if you had a forest fire, maybe the effect wouldn't be that severe. However, as you guys know, you also have to meet these other conditions. So what are bottleneck effects? Hurricanes, tornadoes, right? You name a natural disaster, meteorite collisions, volcano eruptions, tsunamis, right? Am I missing any natural disasters? Terrible floods, hurricanes. I think I said tornadoes already, right? Forest fires, meteorite collisions. Meteorite collisions are natural disasters. These are bottleneck effects. These are bottleneck events that will have a bottleneck effect. And the effect is a reduction in the genetic variation of the gene pool. If the forest fire wipes out 20% of our squirrels, we just lost 20% of our gene pool, and we lost it at random. And it's going to cause the genetic variation to be reduced. Can anybody think of another? Have I named most of the natural disasters?
Forest fires.
Earthquake.
Earthquakes, thank you. Boy, how could I forget that one? Who said that?
I did, Morgan.
Very good, Morgan. Yes, earthquakes. Yeah, and that was the earthquake is what causes the tsunami, right? So bottleneck events lead to the bottleneck effect on a gene pool. And these are random changes in the fluctuation of allele frequencies. In this lab, you guys were going to do a type of genetic drift called the founder effect. Here's the founder effect. Let's say that our population of squirrels, some members decide to disperse from that population. They migrate away. Let's say our population of squirrels has 1,000 individuals, and 10 of them leave the population. The parental population now has 990 members, and this small migratory group of 10 squirrels, they leave the population and go somewhere else. They are called a founder population. That group of 10 squirrels leaves the population, they migrate to another area. They're now far from the original population. Who do you think they're going to have sex with? Are they going to get on little squirrel airplanes and fly back to the parent population? No. The 10 little squirrels have migrated away from the parent population into a different environment, and they're not going to go back to mate with the parents, the parent population. They're going to mate with each other. This is called the founder effect because it starts to concentrate the genes of just those 10 members. In a founder population, you have just a few members starting off. You have a small population. And this is conducive for very rapid microevolutionary change. In fact, the minute the 10 squirrels leave the population, they are taking with them a small subset of the total collection of genes. If they migrate far enough away, which happens in nature, they're not going to be mating with the original population members. They're going to mate within their little small group. That little small group of migratory squirrels is called a founding population. This happens on this planet with all forms of life. There's a tendency for small groups of all forms of life to leave larger populations and to go off and mate with each other. What we're saying is that simple event can cause microevolutionary change. We call it the founder effect. Essentially, the founder effect and the bottleneck effect are going to have the same consequences to a gene pool. You might say, what are those consequences? Well, founder populations are going to have a small gene pool because there were just 10 squirrels that went off with each other. Hopefully they weren't just 10 males because now they're in trouble. Hopefully we had some females in there, right? So they could mate with each other and make babies. So that founder population, just like a bottleneck effect, you're going to get a decrease in the genetic variation of the population. So the founder effect is a consequence of a natural disaster or migration.
Natural disaster.
Founder effect.
Oh, you said founder effect.
Migration.
Yeah, that's the migration. Yeah.
Right. While the bottleneck effect is a consequence of a what?
Natural disaster.
Natural disaster.
Correct. And I promise you guys, on the next 2 tests, you're going to have to discern between these things. I'm going to have test questions that require you to figure out whether it's the bottleneck effect or the founder effect. And the simplest difference is the founder effect is a result of migration, while the bottleneck effect is a consequence of the natural disaster. As you prepare for lecture exam 1, pretend it's going to be the toughest test you've ever had in your life. And then if it isn't, be happy. If it turns out to be the toughest test you've ever had in your life, don't give up on the course. You'll find that a lot of the stuff we're learning for exam 1 will flow into exam 2, and I see much better performance on lecture exam 2, typically. So don't get discouraged, but boy, if you guys don't study for lecture exam 1, it's not going to be pretty.
Is this going to be on the lab exam 1?
This material we're doing right now is on lecture exam 1 and lab exam 1. Both. I guess that's a good thing, is what you're learning, what you'll learn to get ready for lecture exam 1 will help you for lab exam 1. Yep. But I'm just telling you guys, you want to prepare for lecture exam 1 that's coming a week from this Friday, like you've never prepared for a test in your life. Study, study, and study some more. Pretend it's going to be the toughest test you've ever taken. And then hopefully it isn't.
And you said the first 2 chapters of Into the Jungle were going to be like referenced in there, right?
Well, there are elements of you guys don't have to be reading Into the Jungle to be prepared for lecture exam 1 or lab exam 1. It will just supplement it.
Okay.
So does that make sense?
Yeah.
Anytime, if you ever see a video in there that references the Into the Jungle, then yeah, it'll supplement it, but you don't have to have read Into the Jungle yet. All right, so in this simulation, we set up a situation where we create founder populations. Here we have, so we have a gene for bead color. How many different alleles do we have in this parent population? How many different alleles?
5.
5. So we got a gene that causes bead color. We got green, blue, yellow, pink, and purple. We got 5 different alleles for the gene that causes the bead color. Here they're showing you the parental population. And then here they're showing you the founder population. You guys can do this simulation again at home, and you can generate data. I'm just not going to have you turn it in. So I'm not, I highly encourage you to do the simulations. If you follow the directions for this simulation, you're going to see which population. So which population? Parent population or the founder population is going to be the larger one. The larger gene. Yeah, the parent population is going to be the larger gene pool. So when you do this simulation here, you're going to see, here's what happens. You're going to see the allele frequency changes in the parent population to be rather small. It might go 20%, 18%, 20% to 22%. You'll see small fluctuations. Down here in the founder population, huge fluctuations. What might have been 20% green allele in the parent might go to 0% in the founder population. What is 20% blue might go to 50% in the founder population. Because the founder population is a smaller population, it sees more drastic random fluctuations in the alleles. And you'll see that. I encourage you guys to do the simulations. Again, get whatever you like to eat, or just cut up different colored pieces of paper and do the simulation. But this is why trail mix works so well. You could do M&M, you could do some of the M&Ms for one color, you could do peanuts for another, you got cashews for another, and then you got the raisins for this one. I mean, there's probably 5 different things in trail mix. I should have brought my trail mix with me. I love trail mix. So what you'll see when you do the simulations is small changes in the parental population gene pool because it's larger, and huge fluctuations in the allelic distributions in the founder population. So founder populations are going to undergo rapid microevolutionary change. And here's the point. If your founder population migrated far enough from the parent population, they're not going to be doing any gene flow. So let's pretend we got a founder population that's moved over the hill. They've migrated far enough that they're not in close proximity with the parent population. Well, guess what? That means that the founder population is going to evolve microevolutionary divergence over time. You say, why is that? Because in the founder population, there's going to be mutations that occur that won't be shared with the parent population. The founder population might have 50% of its allele frequency for the bead color gene could be blue, while the parent population's like at 18%. Huge differences. Natural selection is not random. Natural selection, how do you think natural selection can cause more rapid change in a large or small gene pool?
Small gene pool.
A small gene pool, yeah. I guess kind of like saying, you know, a large, who could make more rapid changes in their company, a large company or a small company, right? A small company of 20 individuals could make really rapid change in their policies and structure versus a large company of 1 million people is going to take a little bit of time. So founder populations or small populations, they've undergone a reduction in genetic diversity because they've only taken so many of the genes, a subset of the genes from the parental population. And this founder population, if they've migrated far enough from the parent population, natural selection is going to affect them differently. Mutations are going to crop up in the founder population that won't be shared with the parental population. Mate selection, right? You might have a situation now where maybe females in the parental population might have had a certain preference, but now you're in a founder population and your selection is reduced. They might make different choices. If your choice is not to mate at all, right? And all living forms are biologically driven to mate. The female might make a different choice in the larger population versus the smaller population. She may mate with a male that she's not entirely confident is a great choice to mate with, but she will do so because of the circumstances. Questions about founder populations? If you guys haven't watched the videos on the population genetics, you need to do so. If you're not, it'll help you do these simulations, which I encourage you to do so. But I'm not going to have you turn in the graphs or the data. So that means some people will opt not to do it. But remember, I mean, you know, you guys are the ones that signed up for the course. Do you want to learn or don't you want to learn? There's a reason why the majority of people in this country do not have college degrees. Some people just don't want to do the work. So try to motivate yourself to do the work. Questions about the founder effect?
talk a lot about that when we talk about the out of Africa theory. We talked about the different hominid species.
Correct.
Yeah.
Right. A lot of that migration of early humans caused a number of founder effects, which is exactly why you brought it up, Chase, is something we'll deal with later in the course. But that's exactly why, if you look, many people are confused by this, but if you look at the gene, if you define a gene pool as the American gene pool, let's look at the gene pool of Americans. And we compare that to the gene pool of Africans. The African gene pool has more genetic diversity than the American gene pool. People are confused by that because it's like, oh, well, America's the melting pot of humanity. We got such diversity of cultures and such diversity of this and that. Well, what the way we're defining that is we're looking at phenotypes, and often the variations we see in human phenotypes are due to very small genotypic differences. The American gene pool actually has less total genetic variation than the African gene pool. And the reason for that is simple. The African gene pool is humanity's parental gene pool, while all Americans have descended from migratory groups. So some people can trace to European ancestry, some people can trace to Asian ancestry, or whatever. But the people that are in America have a history of migration. And if you follow that history of migration, it all traces back to Africa about 300,000 years ago. So the African gene pool actually has better genetic variation than the American gene pool, because the African gene pool is the parental gene pool of all humanity, where Americans are a consequence, they're a consequence of many, many, many generations of migrations and the founder effects. Does that make sense? So people are very confused on that, because they look at America and they just look at the phenotypes. They go, oh, we got all this variation.
That's interesting, Professor, because as I understand it, it's a lot of the genes we know, we can look at the letters, we can look at the paragraphs, we can look at the sentences, and we can see the differences. But a lot of times we don't know what those genes do. So what are they measuring the genetic diversity by? It's my question.
By allelic variation. They're going to the genes, and if you go into the African gene pool and you find a gene, they will have 10 allelic versions, while the American gene pool has got 3. So there's all kinds of situations of looking at genes, and the pattern is clear. This is not something that's debatable in science. Our home parental gene pool is Africa, and it's clear from the genetic analysis of it, and it comes down to looking at allelic diversity. And so they'll pick one gene, they'll compare, figure it out what the allelic variation is in the African gene pool compared to the American gene pool, and the American gene pool consistently has fewer alleles. You can see it with body height. If you go and look at the distribution of height and compare it to Africa, you'll see that the African populations have much more interesting variation. They got some populations that are really short. They got other populations that are really tall. We don't, the American population is more homogeneous in a way. And it's all, the American population's genetic variation being reduced is due to the founder effect. More on that later. So, but that is one of the major themes of this course. we humans need to learn to work together because we're all related. We all have African ancestry. Try to tell that to about 40% of this American culture. I don't even want to talk about the disgusting nature of our country right now. But we are all of African descent. And we need to respect all of humanity because we're all related, and we need to find a way to come together as a group. Gonna be tough because ignorance is so bad. Scientific ignorance is high. And people want to just, they don't want to accept the fact that we are related to each other as humans and related to the other creatures on the planet. Questions about the founder effect? All right. Now, I have a question for you. If you have a small group of squirrels migrate from a population, do you think the effect on the parental population's loss of genetic variation, do you think it's selected for or by chance? Meaning, do you think those squirrels look at each other and then look around the population and say, oh, I'm really fast at reacting to these predators. I'm going to come with you. This one over here has got a different fur color. Let's make sure he comes with us. Here, the female over there, I like how that tail is shaped. Let's make sure she comes with us. Do you think squirrels do that?
Nope.
No. What I'm trying to say is that those 10 members of this founder population of squirrels, when they left, they're not thinking about genes. They don't have a clue. They just happen to be whatever, friendly with each other, liked each other, and those 10 left the population. My point is the founder effect is chance fluctuation in allele frequencies. It's not selection. The bottleneck effect is genetic drift. It's chance fluctuations. The founder effect is a form of genetic drift. Remember, when you hear the word drift, think chance. Now, this founder population, when they leave the parental population and they get themselves into a new environment, they are often at risk of dying. So it's an interesting balance between the fact that you have this small population in a new environment. At the same time, that small population is potentially able to undergo rapid microevolution. It's also in a situation where the members may just die out. So it's an interesting balance of whether or not they're going to survive and be able to adapt to the new environment. So at the same time, the loss of genetic variation in the small population puts them at risk of dying out. At the same time, they also have an increased chance of rapid microevolution that might adapt them to the new environment. Ready to move on?
Yep.
All right. So we can't predict which members are going to leave a population. So gene flow is random. We can't predict which members are going to leave a population, either emigration or immigration. Exit or entering. To come in, right?
Woo!
So gene flow is chance. We already said that the founder effect is a type of genetic drift, which is chance. Natural selection, however, is not chance. You guys should understand that by now because you should have done a number of examples of natural selection already, I think. If not, you're going to get more in this chapter. Anytime you see the word selection, it's not by chance. In this simulation, what they've done in this simulation, we're going to pretend that we have a dominant and a recessive allele. And the red allele is going to be the recessive one. The yellow allele is going to be the dominant allele. The yellow allele is going to produce a normal protein, while the red allele is the mutated, altered allele that can potentially form incorrectly shaped proteins. So in this case, our representation is that we have a gene that makes a critically important protein. And as long as you don't inherit two of the recessive alleles, so as long as you're not homozygous red, you'll be able to live. So this is a case of complete dominance. So individuals who are born with two yellow beads or two yellow alleles, they'll make a normal protein, they'll be in good shape. If an individual is heterozygous and they have a yellow and a red allele, that yellow allele, because it's complete dominance inheritance, will be enough to make enough correctly shaped of proteins of this particularly important protein. However, if any of the babies are born and inherit two copies of the recessive allele, they will die. In this simulation, you're going to see what happens over the course of time as a result of the individuals who are inheriting two copies of the red allele are dying and being selected against. Here's what I'm going to do. I want you guys to work through this simulation. I think I'm going to wrap up here and go to questions and save this for Friday. I'm going to go through the details about simulation 3 on Friday. And then I'm going to get into the Hardy-Weinberg equilibrium or Hardy-Weinberg equation. So you guys should go through, here's what you guys need to do before Friday. You should work through these simulations, and you should do the natural selection one. Why is it selection? Because we're going to see that those individuals born with the homozygous condition of two red alleles are going to die. They're going to be selected against. And whenever you have natural selection, you can make predictions about what's going to happen with the allele frequencies. But I'm going to discuss the details of this and answer these questions here on Friday. You guys should prepare yourself by watch this YouTube video.
Hello, everyone. I have something to ask you. Will you add your name to join my campaign? We still have a long way to go if we're going to defeat.
Guys,
Hi, it's Mr. Anderson, and in this podcast, I'm going to hopefully show you how to solve Hardy-Weinberg problems.
I will just simply say this. I've not randomly quickly selected these videos. I've taken a lot of time to pick out videos that I feel do as good a job as I would have done or a better job. In some cases, it takes me a while to select these things because I'm looking through a lot of videos. So if I put up a video here, this isn't for just for, it's recommended. No, if you guys want to learn about Hardy-Weinberg, you better be watching them. So by Friday, you guys want to move through these videos here about Hardy-Weinberg, and then we will discuss the Hardy-Weinberg equation. And we got some other stuff to discuss as well. But I'll finish the natural selection one on Friday. Let me go ahead, before we go to questions, let me go ahead and do this one. What are the chances of you guys seeing these graphs on your lab exam, do you think?
High.
Pretty high.
100%. I've finished the lab exam. It took me a lot of time, and these graphs are in there. Here's my question to you. The graphs represent genetic drift. One of them is genetic drift in a small gene pool. One of them is genetic drift in a large gene pool. When you guys see the word genetic drift, you should look at drift and think what? Selection or random?
Random.
Random. By chance. The genes are drifting in random ways. It's not natural selection. But if, look at graph A here. You start off with an allele frequency of about 50%. Notice it's showing you the different generations here. Which axis is the x-axis here? Percent alleles or generation?
Generation.
Generation.
Right, so on the x-axis, we got the generations, 20 generations, 40, 60. On the y-axis, we have the percent alleles. Notice we start off at generation 0 at about 50% allele frequency. And notice how it goes up, goes down, starts to go back up, draws a little bit different. Now, compare that to graph B. It goes up, comes down, goes up, comes down. Which of these graphs do you think is the small gene pool? B or A?
A.
A.
A. Why? Because genetic drift is going to have a much more pronounced effect on a smaller gene pool. Small gene pools can undergo more rapid microevolutionary change. This is the large gene pool here being affected by genetic drift. These are random changes in the allele frequency of a large, so B is the larger gene pool, while A is the what? Smaller gene pool.
Is there ever a situation? Yeah. Is there ever a situation where it goes up? I'm just trying to think of like if there's ever.
Like here in graph A?
Well, just in general, just would the gene pool ever go up? You know, like the variations.
Yeah. Well, if you say increase in variation, yeah. That could, yes, I'll just say yes.
Okay, thanks.
Yeah, yeah, I don't want you to think that just because this ended up going down, that you can't have situations in which it could end up going up.
Okay.
Right? As far as the percent alleles. Does that make sense? So yeah, you can have an increase in genetic variation that happens over time as populations get larger due to mutation. It can also happen as a result of gene flow from neighboring populations.
So adding on to Morgan's question, like for graph A, if it's going up, does that mean like a positive genetic drift happened, and then if it's going down, it's like something bad happened?
Yeah, no, we don't use those terms. We would just say that here, the allele frequency went up.
Okay.
Here it went down. Here it went back up, then it's going down. Here it goes up. You with me? It depends on what point you're looking at.
Okay.
If you're starting it, and I might ask you to see if you can read a graph, right? And if I say, hey, what is the allele frequency at 80 generations? You go to 80 generations, you go straight up from there, hit the point on the graph, and then straight over from there. You guys with me?
Okay.
Everybody understand how to read the graph. If I ask you what's going on at 40 generations, you go 40 generations, up here, intersect, right there, boom.
Yeah, you're saying if the alleles percentage is going up, you have to also ask which alleles is it, because if it's going up, and it's definitely getting selected for.
No, in this case, it's drift, so it's not being selected for. What we're saying is the reason it increased was due to some random aspect in the gene pool.
Yeah,
it wasn't.
Yeah, but I'm saying if an allele in a gene pool is getting more frequent in a gene pool, then it has to be getting selected for within the population. I mean, where else would it be more frequent?
Because you can have, it can happen because of drift. So listen, this is how it happens. Let's say that we have our large population. We got our squirrels, and a couple of squirrels have some funny tails. In the large population, very few of the alleles make funny tails. But let's say that a few of those squirrels with funny tails leave the population to form a founder population. That's drift. And now the only people they can mate with are with other squirrels with funny tails. Now the allele frequency increases. It increased simply not because of selection, but just because of drift. It was by chance that those members that formed that founder population happen to have those alleles.
Okay, so it's increasing for that specific trait because that's the only thing that's in the population.
Correct.
Okay.
And that was due to drift.
Yeah, that's sorry.
That was due to the founder population. Yeah.
Yeah, but I was just saying that a drift doesn't have to be the only thing that can increase the alleles percentages. I mean,
no.
If an allele is the allele,
you didn't say it that way though. You did not say it. If you'd have said it that way, then that's fine. You're right.
Yeah,
drift,
that's what I meant. Yeah, I wasn't.
Okay, well, that's not what you said.
Yeah,
you're right. Natural selection can cause alleles to increase. Yeah, but drift can do it too. Okay. Yeah. We're good. Can everybody read these two graphs? B is what? Small population or big one?
Big.
Large.
Yep. All right. Yes, but you're right, Chase. Other factors can cause allele frequencies to go up, and selection can be involved, but drift can also be involved.
Oh, definitely.
Let's go to one more graph before we. What do you think of the chances of seeing this on your lab exam are?
100%.
100%. First of all, just make sure, again, you get the y-axis with percent alleles. You got the generation here. In this case, we're showing you two different alleles on the same graph. While the other case, it would look like it was just one allele. They're showing you a dominant is in the blue, recessive in the red. And if we, in this particular case, we're going to assume we just have two alleles for this gene. Notice if you have just two alleles for the gene, notice it makes sense that if the recessive allele is at, let's say, 35% frequency, then the dominant allele needs to be at 65% frequency. Does that make sense? Because these represent the only two alleles for that gene. And the totality, when you add up, if you only have two alleles, adding those up, the frequencies of those has to get to 100% of the allele frequency for that gene. Has to add up to 1. You guys with me? When we do Hardy-Weinberg on Friday, you're going to find out that the P symbol's for the dominant allele, while the Q symbol is for the recessive allele. More on that on Friday.